everyone, welcome to this class of cell signaling course. This is a summary class where we are going to see what we have learnt during the course, and I'll be presenting a snapshot of cell signaling. In brief, I'll be saying what we have understood in this course, why signaling is required, what all is required for signaling, what is the purpose, what it ends up being as part of the end result of signaling. So it is very poorly appreciated that life starts with a signaling event, where fertilization process, when ovum and sperm come together to give rise to zygote. This is very much a chemo-attraction type of signaling which happens. Once this process has started, then every aspect of this zygote getting into a individual is regulated by signaling. So what is the requirement of the signaling? What is signaling? The development aspect, as I said, from the zygote to all the way to the individual, grown-up individual or newborn, the organism needs multiple type of signaling. Why? Because organism is growing in number of cells, tissues, diversity and so on. So this message has to be communicated that differentiation of a particular type of tissue should happen, a cell should become other cell type and so on. So the signaling which will be present in multicellular organism will be of different types, which sometimes may not be present in the unicellular organism. And all these multiple signaling events must be coordinated very well. Otherwise, it could lead to a developmental issues and later form a bigger defect thing. Functioning of different organs, they need signaling, communication between them, and that is where we know that how hormones play a very important role in this communication. And we have seen that how a particular type of receptors, they do engage in hormones binding and the signaling. Signaling generally, although seen as a macroscopic view happening between maybe one organ to the other organ or one tissue or to the other tissue, but microscopically it is happening between the two cells. And these two cells, one of them is called signal producing cell and the other one is signal receiving cell. We understood that the signal is made by the signal producing cell and this is sensed by the receptor on the signal receiving cell, either present on the plasma membrane or it is present inside the cytosol, or it could be present inside the nucleus. This combination of receptor and signal is one of the first mandatory event for signaling to proceed forward. So the signaling could be of multiple different types. It could be short range signaling, which means that signal which is produced travels a smaller distance. And in case of long range signaling, the signal travels along with blood circulation to far reaching distances in the body, in the multicellular organism. This, in case of short range signaling, signal is delivered locally or generated locally, whereas long range signaling, signal is produced, goes into blood circulation and goes to the longer distance. The way these signals work defines the signaling type. The paracrine signaling, meaning the cell which is making signal and the cell which is receiving signal are different. So if I say signal producing cell and signal receiving cell, these are distinct. They could be present in closer proximity or they could be present at far off distance. So the long range signaling is a kind of paracrine signaling. The autocrine signaling, here the signal producing cell and signal receiving cell, they are same. Basically a short range signaling could be a type of autocrine signaling as well, or there could be juxtacrine signaling where you would need the signaling to happen in close proximity and may involve both signal producing cell and signal receiving cells to express receptor and ligand and get into cell-cell contact mediated signaling. So basically you want signal producing cell and signal receiving cells to be juxtaposed in this form of signaling. What is the need for the signaling, or rather what all is required to do the signaling? We have already said that signal is one of the very first thing which is required. The receiver of that signal, the receptor, and then something which is effector or transducer, which sends the signaling inside the receiving cell. And we have seen that signal could be soluble or membrane bound. Similarly, receptor could be soluble or membrane bound, and they are proteinaceous in nature. If you think about their biomolecular nature, they are proteins. But in case of signal, it could be anything. It could be as abstract as a photon. It could be DNA, it could be protein or peptides or amino acids, it could be lipid derivatives or sugars. So the most diversity in terms of biomolecule exists with signal, because any possible biomolecule metabolite that can serve as a signal. However, the receptor which we are talking about, they are always a protein entity. The effector which is generated inside the cell, sometimes messengers or second messengers we have seen in some signaling, they are the transducer, but largely you will see again proteins which are your effector molecules, which could have a adapter function or a scaffold function and many more things. The signaling also needs regulation, because we understand that this is required for many aspects of the organism's growth. So signaling needs regulation, for which every component is regulated, and one of the simplest way is to modify them with a small group like phosphate. So we notice how phosphorylation, dephosphorylation played very crucial role in signaling and activating the process. We also saw some small GTPase like molecule which came together. So this phosphatase or phosphate group addition and removal worked very well for all these molecules, whether it is signal or the receptor or the effector. GTPase were one of the effector molecules which switched between on state and off state. That is how they communicated the signaling. As well as in case of signal, we saw how signal was produced as a precursor, or for signal and receptor we saw, or effector we saw they were bound with inhibitor, or signal was produced as low half life, and effectors were localized to one place versus another place. They could be nuclear when activated otherwise they are cytosolic, where they can still be bound with something which inhibits their activity, or they are phosphorylated, or for that matter they could be anything, like we saw in case of cyclic AMP, GMP and cyclic di-GMP in case of animals. So their cyclic form is something which is important, and their linear form was something which is not important. Now if we think about the signal, as we said, they could be soluble or secreted, or they can be as active molecule. Basically you have something like produced as a precursor, this pro domain is cut and released, and this generates the active molecule. Sometimes they are produced as a molecule which needs the conversion. Basically you saw the testosterone and its active form, which was dihydrotestosterone, or for that matter ecdysone or 20-hydroxy ecdysone. So they could be having multiple different types of regulation for generating the active molecule. They could be secreted but protected because they are in hydrophilic environment and signal is hydrophobic. Or they need to be compartmentalized. They are protected in the sense they are packaged in vesicles like neurotransmitter. Thus, they can also be membrane bound and be localized to a particular place. We saw something which was present in the plasma membrane, something could be present in the ER membrane or in other membranes which are part of organelles inside the cell. So signals could be of multiple different types and they could be regulated by multiple different means. If you talk about the receptors, they are generally membrane bound. Large portion of the receptor which we saw, they are either tethered to the plasma membrane, which is again the most abundant type, and they could be organelle bound, which is like ER membrane, or they could be in the mitochondrial membrane. So there are multiple different receptors, but largely you have seen that they are in the plasma membrane. If you recollect, in case of ethylene receptor, they were ER bound. But all other receptors which we saw, whether it was receptor histidine kinase, receptor tyrosine kinase, receptor serine threonine kinase, or for that matter GPCRs, they were largely the plasma membrane bound. Then there are soluble or cytosolic receptors. We saw that how in the class of nuclear receptors, we had cytosolic in case of ecdysone, and ligand binding was taking it into the nucleus and activating it, and in case of thyroid or androgen and so on, they were already in the nucleus and binding of this ligand was activating them and switching them from a co-repressor bound state to a co-activator bound state, and transcription is turned on. The receptors can be of the type where they have integral enzyme activity, where they can come with kinase activity or phosphatase activity. They can be there with the phosphatase activity or kinase activity. We have seen these activities are driven towards particular amino acid or even to the other biomolecules. We know that there are, if you think about the protein kinases, there are histidine, serine and threonine, and tyrosine. We also saw some examples of aspartate receiving the phosphate group, and those were aspartate kinase. We have seen phosphatase activity for all these molecules, whether it is tyrosine, histidine or serine threonine. We also saw some other enzymatic activity associated with the receptors, but there were enzymes, there were receptors which largely dependent on the binding of an enzyme activity bearing molecule. We saw JAK-STAT pathway, where JAK is the kinase which associates with the receptor. The receptor does not have the kinase activity. We saw a large class of receptors in animals, the GPCRs. They bound with the G alpha, which is a GTPase, and it also had integral GEF activity. Then you can think about the receptors which have transcription factor activity. All your nuclear receptors, they by default had this transcription factor activity because they carried a DNA binding domain. In many other cases, we saw effector carrying some of these transcription factor activity and DNA binding domain regions. But in case of nuclear receptors, we saw that how DNA binding ability was there integral to the receptor, which either bound to palindromic sequences in the DNA element, or it bound to the tandem sequences in the DNA element. We saw it was binding to the duplicate of tandem sequences, or it could bound to extended sequences, and this binding was helping in the transcription process. In many of the cases, you would have noticed that they were involved in transcription related activity regulation. Then we saw that there are receptors which are self-transducing type, which means that they are mostly channel type. They were not dependent on the effector molecule. So if the channel is closed for any region, the signal opens them and they allow the movement of ions. So the receptor itself was serving to communicate the outside message to the interior of the cell, thus it was a self-transducing type receptor. Then we saw the effector molecules which come in different flavor, and these are the key players in directing the signaling in a given direction. They came with a enzymatic activity. In case of JAK we saw, in case of many other kinases we saw like PKA, PKB. We also saw how PI3 kinases were involved. We also saw how lipase activity was involved, and so on. We saw some of these receptors which had the phosphatase activity. We saw some of these effectors which had the phosphatase activity, and they removed the phosphoserine, phosphotyrosine and phosphothreonine so that they can be activated or inactivated, or the equilibrium between the active and inactive state could be maintained. Then we saw some of these receptors with the ATPase activity in case of microbial world, when we saw different response regulators. They had this ATPase activity. Also in case of these self signaling or transducing type of receptors, if you think about sodium potassium ATPase, that allowed the movement of ion at the expense of ATP molecule. We saw these effectors coming with the cyclase activity, whether it was adenylyl cyclase, guanylyl cyclase, or diguanylyl cyclase activity, which allowed ATP to go into cyclic AMP or GTP to cyclic GMP. And then we saw these phosphodiesterase activity working on them, getting them converted into AMP and GMP like molecule. We saw how cyclic di-GMP was first produced using the GTP and then broken down into GMP, GMP dimer, and then finally to a single GMP molecule. We also noticed that many of the effectors had the transcription factor activity, for which they had one of the domain to recognize receptor and then the domain which has the DNA binding and activation domain, so that they could serve as a transcription factor, and then binding of this allowed the transcription to continue. We saw effector molecules as a scaffold or adapter, in the sense that you have the receptor which generates a residue on which the next molecule will come and it will activate something. So, we saw there here some of the GRB2 like molecule in case of receptor tyrosine kinase activity, they were activating the RAS pathway, the MAP kinase pathway. Or you saw the receptors which generated these sequences which recruited a molecule which was the scaffold. The scaffold now will recruit next molecule to take the signaling forward. So, we need this cell signaling, or why does cell signal? What all it has to meet? So, in essence, as a summary, very succinct summary of this course is that cell signals to communicate with its surrounding. If there is a neighboring cell, we saw in case of bacteria, bioluminescence, biofilm formation. But communication is important so that cell can survive and perform its function efficiently. It does signaling so that it can divide and grow in number. The cell signals so that it can differentiate from a progenitor to a differentiated cell. We saw how TGF beta and BMP signaling was important to change the osteoprogenitor to osteoblasts and osteocytes. The cell signals to survive, because there could be a signal which will be apoptotic signal, or there could be death associated molecular patterns which can cause problem, or there could be infection. To survive all those things, cell signals so that it can come to terms and maintain the homeostasis with the stress or the signal which has been put up over there. Last but not the least, these cells do signal to migrate from one location to other, normally chemoattraction, immune cells, they will signal and they will move to a new location, they will respond to signal and move to a new location. But in case of disease like cancer, you will have EMT, where cells migrate from one location to other location and establish the new location for cancer progression through this metastasis process. So in essence, we have understood why cell signals, what is the need of the signal, what is the requirement for signaling, how they are regulated, and how they are coordinated to ensure that cell is surviving, proliferating, doubling, responding to the environmental cues, and communicating in very efficient manner. Thank you.